Good afternoon. I know it's good news, but I hope everyone grabbed some coffee because it's late afternoon for me. Today I wanted to talk about a project that I've been working on with two of my colleagues from the Plumas National Forest, Kyle Marion and Jim Belcher-Howe, for I think a little longer than we ever anticipated when we started it. And although this particular story is about one specific species and the particular actions that we have taken to restore it, I really do hope that some of the lessons that we've learned and the process that we've gone through can resonate with some of you out there in the audience, particularly those that work with some of these rare species, and when monitoring informs us that action might be necessary, having the courage and knowledge to actually take some action and work on bringing some of these species back from the brink. So one step forward, two steps back. I'm going to be talking about some of the work that we've done with this species. This is the star of our show, Astragalus webberi, Weber's milk-vetch. It's a beautiful perennial in the pea family, has showy yellow flowers. It's one of our more charismatic rare plants that we have on the Plumas. And the California Native Plant Society ranks it as 1B.2, so it's a pretty rare plant here in California. And if we look at its distribution, we can really see how rare it really is. It's endemic to central Plumas County, which is just east of where we are today, and it's limited to 13 occurrences. And if you look at this map, the little colored circles show the complete extent of this species. And so if you look from the easternmost to westernmost population, that's a distance of about 25 miles. So we're talking about a really, really narrow distribution for this plant. And also, one of the other things that really stuck out to us is that about half of these occurrences are on private lands. The remaining half occur on public lands managed by the U.S. Forest Service, and that's where we can concentrate most of our restoration actions. So back in 2003, Jim Belcher-Howe, a botanist on the Mount Huff Ranger District of the Plumas, undertook this survey effort to revisit as many populations as he could. A lot of these hadn't been revisited since the early 1980s, and so he went back and he looked, and what he found was somewhat alarming. A lot of our populations were in pretty steep decline. They declined by as much as 22% over this 20-year period. And when he tallied up all of the individuals that he had counted, we were looking at about 2,000 plants covering less than 10 acres. And when we started to look a little more carefully about where these plants actually were on the landscape, they were in a pretty precarious place. A lot of them were growing on these steep cut slopes next to interstate highways or Forest Service roads where they were really easily impacted by road maintenance activities. And for those occurrences that were not close to roads, they were limited to these really small gaps, oftentimes surrounded by pretty dense canopy conifer forest. So when we took all of this into consideration, the declining numbers, where they were occurring, kind of these precarious places on the landscape, we decided that we really needed to take action. But like so many of us, we don't really know a lot about the biology or ecology of this species, so we wanted to take a somewhat cautious approach. So we decided to take two aspects. One, we would focus on expanding some of the existing populations that we had through some of the restoration tools that we have at our exposure, and then we wanted to really establish new populations. So in order to approach our first objective, which was to expand existing populations, we focused in on one particular occurrence. And this is one that we like to call the China grade occurrence. It occurs just south of Taylorsville, for those of you who know your Indian Valley geography. And this particular occurrence is one of our largest. And you can see that the large polygon in yellow represents a pretty big area of plants that is mostly on private land. And when we were doing our surveys and we were peering over the private land boundary, we didn't go on private land, of course, but when we were peering over, we noticed that there had been a number of treatments that had been implemented on this land, and that these plants were doing really, really well. So we thought, well, maybe we could try to implement some of these restoration treatments on the Forest Service portion of this occurrence. So we focused on two different questions. First, we wanted to know if we could expand an existing population that we had through some of the restoration tools that we commonly use for forest restoration, things like thinning and prescribed fire. And the second part that we really wanted to know was, can we expand these populations by actually planting seeds and seedlings that we had grown in the greenhouse? So we separated our occurrence into two units, one that we would leave untreated, our controls, and then the remainder that we would actually implement through a series of treatments, which we would monitor at each phase. So the first one was we did a hand thinning and piling project in 2008. And these pictures, I think, really show a great example of what this stand looked like, or what this occurrence looked like prior to treatment and what it looked like after. And the aerial photographs actually show as well how just by hand thinning small diameter trees and piling the material, we were really able to open up this stand and increase the amount of light that reached the understory. And then in 2009, we burned those piles, and importantly to our story, we started preparing for our prescribed fire. And what that meant is we really wanted to minimize the direct impacts to some of our existing plants. So we actually dug fire line around small clusters of plants. And then in the fall of 2010, we burned the site, and you can see a great example of that control line that I'm talking about, right up there in the upper right, where you can see that it's actually pretty successful at keeping fire out of those existing clusters of plants. So our question, can we expand this population through fire and thinning? And what we really focused on was tracking individual plants in both our treatments and our controls. And what we found was really great. Plant numbers increased significantly after treatment. This graph shows the number of plants on the y-axis and the time steps that we monitored along the x. And the bars represent the number of plants in each of these different size groups. So the red here is some of our larger plants, and the kind of yellow-green are more of our seedling cohort. And so what you can see is that over the life of this project, over the 7 years, we saw plants increasing from less than 30 prior to treatment all the way over to 400, more than 400 after treatment. So this was a really successful treatment compared to our controls where we saw plant numbers remaining pretty stable over time. And one thing that was really interesting to us is that during our treatment, we saw a pretty big increase in that time period where we had piled our materials and put in fire line, but before we burned. So we were really interested in understanding what exactly the mechanism was there that was really stimulating germination of all these seedlings. So we took a look a little more carefully at what was happening within our monitoring grid. This schematic on the bottom shows our monitoring grid, and the green points are where we estimated plants to be prior to treatments back in 2008. And on the right, you can see that same graph that I just showed that correlates to how many plants were actually in that monitoring grid at the time we were sampling. And if we fast forward 7 years to 2015, what we can see in the purple are where plants are today. And we can see this huge increase in the number of plants in that kind of northwest corner of our monitoring grid. And here you can also see in the orange was the actual area that was burned in our monitoring grid. So that was really surprising to us because we weren't seeing this large increase in areas that we burned, but we were seeing in areas that we had excluded from fire. And when we looked closely, you can see in those red arrows, it shows kind of where the corners of the plots are in both the photo and in the diagram. You can see that this area was pretty highly disturbed. And that when we looked carefully, we saw that most of the seedlings that we were finding were actually coming up in this fire line. And I think I need to mention here that Astragalus webberi, like so many species in this family, has a long-lived, hard seed coat. So it needs mechanical scarification of some sort to actually stimulate germination. So what we thought is maybe actually by scraping the duff and litter away from and exposing bare mineral ground, they were actually being able to stimulate germination of this rare species. The other cool thing that we found was that growth and reproduction of some of the plants that we had tagged and tracked over time increased significantly with treatments. So this graph basically shows on the left side the total number of change in the total number of stems, and the graph on the right shows the change in the total number of fruiting and flowering stems. And the blue is the treatment and the red is the control. And it's really clear, even to those people sitting way in the back, I'm sure, that really we saw this pretty significant increase in the size of these plants over time in our treatments and did not find the same increase in our controls. And when we looked a little more carefully about why some of these species might be or individuals might be responding like this, we found that a lot of them occurred in areas with lower overstory canopy and areas with shallower litter. So that led us to think that some of the environmental conditions that we were creating by these treatments were really stimulating growth and reproduction over time. And something else that was really interesting to us is that we had a lot higher survival in our treatments compared to our controls. We lost about half of our plants that we were monitoring on our controls over the life of the project. This is just a great photo series showing what was actually happening in that last graph. On the top, you can see the changes prior to treatment and then after treatment in our treatment plots compared to our controls on the bottom. And you can see this really significant response from these individuals over time in response to some of these treatments that we implemented. Another really cool and very unexpected thing that we saw was that treatments actually stimulated germination from a really long dormant seed bank. When we did these surveys back in 2003 and then consecutively every year during the life of this project, we didn't think that we would actually see very many individuals outside of the known occurrence. But after we implemented our prescribed fire, and then we went back 4 years later, we found 49 new plants growing in this area just north of where the existing population was. And these plants were really big and healthy and flowering, so we're still trying to figure out kind of what it is about this particular site that's actually driving this, growth and germination of these Weber's milk-vetch. So, can we expand the existing populations with thinning and fire? Yes, we think we can. We saw really great results, increased plant growth and reproduction with our treatments, and we also saw a really great response in terms of stimulating germination from a pretty long dormant seed bank. So we're pretty excited about that. So our next question was, could we expand this existing population by planting seeds that we collected from plants in the population and greenhouse-grown seedlings? And to do this, we went through a series of experiments, which I'm not going to go into too much detail, but we basically planted seeds prior to burning, and we also planted a number of seedlings after we had completed all of our treatments. And then we monitored all of these places over time. And what we found is that burning alone, without that kind of ground-disturbing activity that we saw in those treatment units, was not enough to stimulate seed germination. We had really low germination in our seed plots, about 2 to 3%, regardless of the treatment type or the intensity of the fire, which we also monitored. And we saw really low survival. 1 year after the seeds had germinated, we saw about 39% survive to 1 year, and none to 4 years. I know this was supposed to be the good news, but there is a little bit of bad news sprinkled in this one. We also found that the survival of the plants that we planted was what I'm going to call marginal. This graph, or this map here shows the 74 plants that we planted in the unit. They're represented in green circles. 2 years after we planted them, we had pretty good survival. We had about 72% alive. 5 years after planting, that's where I think we have more marginal survival. We had about 18% surviving. But the interesting thing to me here was that most of these plants, 9 out of the 13, were actually 100 feet from that original occurrence, in that same place that we actually saw those new plants. So there's something about this site that's actually driving the survival of these plants. And it's not any of the things that we've measured, like canopy cover, duff and litter, slope aspects, but something else. So if anyone has any ideas, please let me know. So can we expand the existing population by planting seeds and seedlings? We think so. We think we're still trying. After all these years, we're still trying. We need to scarify the seeds prior to planting. We know that now. And we also need to really identify some of the environmental conditions that are regulating the growth and survival of these seedlings. So remember our plan for action. We had 2 steps. We had focused on expanding existing populations, but we also wanted to know if we could actually establish new populations outside the range of what we already know. So we focused on answering a couple questions. One, can we successfully grow these plants in the greenhouse? And once we do, will they survive when we actually outplant them into new sites in the landscape? And this is where we enlisted our local high school students, a really great group of students and teachers from Greenville High School to help us collect and scarify seed from all of the known populations. We planted the seed in the greenhouse in a common garden experimental design. And then the students actually grew the seedlings in the greenhouse and monitored their growth and survival. And then we took those seedlings that we had grown and planted almost over 900 plants into 3 different sites, which we had chosen based on some pretty complex habitat models that we had derived. And then over time, we monitored the growth and survival of these seedlings. And what we found is that if you have a great group of students and teachers like at Greenville High School, we can successfully grow plants. We were able to get pretty high germination rates, 55 to 75%. And this is over a 3-year period. And we worked really hard in kind of honing in on our scarification methods and how we actually get them to grow. But one thing that was really interesting to me is that we tracked how different populations did in the greenhouse and the common garden. And what we found was that 2 populations, the ones from Misery Bar and Pioneer, consistently outperformed the other populations. So that to me is really interesting from a genetic standpoint because it suggests that they might actually have something that's conferring a little bit of an advantage in this common environment. The problem is that we can grow them, but once we plant them, they might not survive. And that's not good news. Survival the first year, again, was pretty good. It was 26 to 38%. And interestingly, those same two populations that did really well in the greenhouse were outperforming the other populations when we planted them in the field. The problem is that survival the second year was 0. So we have one more site that we haven't collected our second year information on, but that was a little depressing, especially for all of us who worked really, really hard. But again, we looked at really what is driving this relationship between survival and some of the environmental conditions of these sites, and there hasn't been a really clear relationship. So we're still trying to work on that. So can we establish new populations? We know that we can actually grow plants in the greenhouse. We have it pretty dialed in now. We're really successful at that. But will they survive in new sites? I think only time and a little more experimentation will really tell. And this is actually a picture of a project that we're starting right now, looking at how we can kind of experimentally alter shade and water to see if those two factors may interact to really influence survival and growth of this species. So what have we learned? If I take a step back and think beyond just Astragalus webberi and Weber's milk-vetch, I think something that might apply to all of us is that sometimes when you have declining numbers and a rare species like this, that you do need to take some action in order to bring it back from the brink. And that really, some of these treatments that we often apply to landscapes in terms of restoration, things like thinning and prescribed fire, can be used in moderation to definitely expand some of these known populations. And this was really cool because here we were able to show that you can even expand them once they've been absent from that landscape for a pretty long time. But really to do this, you kind of always need to think about how you're balancing the risk, right? I mean, you need to balance your direct impacts from treatments with that risk of loss. And to do that, I think it's really good to take kind of the stepwise approach to stop and monitor and learn, even if it takes you 10 years like it has us. But I really do think that if you can learn along the way, then you can really design these treatments to be most effective. And really, this is just kind of a plea to everyone. I think working with rare species can be incredibly challenging and frustrating. We don't know very much about these species. There's not a lot in the literature. But I think that all of these projects that we work on can really help us to learn a little bit at a time and really help us to learn as a group about how we might work to bring some of these species back. And I just really like to thank everyone who's worked on this project, especially all the students and teachers and assistants. Thank you.